Hey guys, so today we're going to be talking about proteins, and essentially what proteins are just molecules found within every single living cell that are necessary for survival. They are a polymer, which are called polypeptides, and each polypeptide is made up of amino acids. So the amino acid is the smallest unit, so to say, for a protein. And the first thing we're going to need to know about an amino acid is how to draw one, or at least how to recognize one. And how I like to do it is by dividing it into three different sections. This group over here, which is called the amine group, and it's made of the nitrogen and then the two hydrogens bonded to it. Then the carboxyl group, which is a carbon double bonded to an oxygen, and then also a hydroxyl group, or a hydroxyl, yeah. And then this middle part, which is the carbon that bonds the amine and the carboxyl group, also bonded to a hydrogen. And then this is the important part, the R group. Now the R group isn't fixed in an amino acid. It differs from every amino acid, and it's what gives it its function. So depending on the R group that amino acid has, makes the function different. Everything else within the amino acid, in every amino acid, stays the same. So the amine and the carboxyl and this carbon in the middle bonded to the hydrogen and all that stays the same. But the R group is what changes. Now there's different types of amino acids found within the human body, and those are essential and non-essential amino acids. Essential amino acids are not produced by the body, and we need to be able to include them in our diet so we can consume them, so we can actually utilize them. And then non-essential amino acids are ones we don't need to consume or include in our diet because we are already producing it with our own body. Now, to create a polypeptide, the two amino acids or more, like 3, 4, 5, however many there need to be for this protein to function, they need to undergo the condensation reaction. Similarly to carbohydrates, how a monosaccharide can bond together with another monosaccharide to create a disaccharide or polysaccharide. The same thing happens here. So through the condensation reaction, we can get a polypeptide chain. So it's polypeptide. And essentially, the bond is called a peptide bond, and it's this part over here. Sorry, you can't really see that, but this is called a peptide bond, and it's the carbon and the nitrogen bonded together, and then we also include the oxygen and the hydrogen because that is the part that changes. So if we take this amino acid, for example, let's just say that this group and this group are on two different amino acids. The hydroxyl group, or the OH, is removed from this part, and one of the hydrogens is also removed from this group, and they break off and produce water, or H2O, because it is a condensation reaction. And then this carbon bonds with this nitrogen, which is how you get that bond right there. So each protein has their own favorable environments. They're all very specific to that specific protein. Not all proteins are the same. And when you change things about that environment, such as the temperature or the pH, that can change the protein as well. So in temperature, as the temperature decreases, you get less protein activity, but that's due to a loss of kinetic energy. But as you increase the temperature, weaker and then stronger bonds can start breaking. So for example, if we take this polypeptide chain, and we see this disulfide chain, which is a weaker bond, that can then be broken as the temperature increases. However, if you keep breaking that bond, the peptide bond between two amino acids, which is a covalent bond and a much stronger bond, will also start to break. And the pH will change the R group's acidity. So it can be, and essentially what this does is, as we said before, the R group is what gives the protein its functionality and also its structure. So when you change the temperature, when you increase the temperature and change the pH, that all causes the protein to lose its shape, and therefore it becomes denatured. And that is when that protein becomes inactive and unable to do the function that it has been set out to do. So for example, if we get an enzyme with a specific active site for a substrate and we put it in temperature that is way too high for it to function in, it can change the shape of the active site, and therefore the substrate can no longer bind to that enzyme, and it can no longer be catalyzed. So yeah, that was it all for proteins. There's really not a lot in my opinion, but yeah, it's very interesting. So I hope you enjoyed, and the whiteboard for this will be added below. Hey guys, so today we're going to be discussing the higher level content of the topic of proteins, which is part of the IB biology curriculum. So essentially what we need to know for higher level is the different structures within a protein, which are primary, secondary, tertiary, and quaternary, and then also two classes of proteins, which are fibrous and globular proteins. So let's get started with the primary structure. Essentially, the primary structure is the first structure, and it's just a chain of amino acids held together through peptide bonds. As we know from standard level, a chain of amino acids has a peptide bonds. And this is the first structure, but it's probably the most important in determining the outcome of that protein, because it determines the sequence of amino acids. Now we'll get to this again, but every single amino acid has a different R group, and those R groups have different characteristics, those being positively charged, they can be negatively charged, they can be polar, and they can be non-polar. And what this does is it can change essentially the function of that protein depending on the sequence of amino acids, but we can talk about that in the tertiary structure later. So then we've got the secondary structure. You've got beta pleated sheets in the secondary structure, and you've got alpha helixes. And essentially these structures are formed through hydrogen bonds, which are essentially interactions between the oxygen of the carboxyl group and the hydrogen of the amine group, of a different amino acid of an amine group. And essentially the reason you get different structures is because in alpha helixes, there is a hydrogen bond between every 4 amino acid. Whereas in a beta pleated sheet, it's kind of the backbone. They use the backbone of the peptide bond to create a hydrogen bond. So if we've got a sheet here, it'll then wrap around and follow that pattern. So these would be the hydrogen bonds. So it just depends on where those hydrogen bonds are found that determine whether it would be a pleated sheet or an alpha helix. Then we have a tertiary structure. The tertiary structure, it's what gives a protein its 3D shape. It's also To get the shape, you do have some hydrogen bonding, as well as other bonds, but the difference between the hydrogen bonding in the secondary versus the tertiary structure is that in the secondary one, it's between the oxygen of the carboxyl group and the hydrogen of a different amine group. But in the tertiary structure, it's the hydrogen bonds between the R groups, so not the general component of the amino acid. It's part of the R group. Other reasons for differences in structure are things called disulfide bonds. This is a covalent bond, and it's between two cysteine amino acids. And the reason it's only because it's through two cysteines is because cysteine is the only amino acid that has a sulfur in it. So the sulfurs of two cysteine amino acids will bond and create a covalent bond between them. Then you also have ionic bonds. So another reason for the shape could be that you have a positively charged R group and a negatively charged R group, and there will be some sort of attraction between those two amino acids, or the R groups in those amino acids, that force them to come together. And then you also have hydrophobic or hydrophilic interactions. So essentially, hydrophobic molecules do not like water, right? And hydrophilic do like water, or more specifically, hydrophobic are nonpolar and hydrophilic are polar molecules. So when you get when you put a protein inside of water, for example, you will get hydrophilic amino acids on the outside of that protein. So if we have, let's say we put this protein inside of water, which is a polar substance, you would get the hydrophilic amino acids on the outside, and then you would get all the hydrophobic amino acids on the inside. Now, if we put that same protein in a nonpolar solvent, the roles would be reversed. You would get the hydrophobic amino acids on the outside, and the hydrophilic amino acids on the inside, trying to stay away from the solvent. So all of these different bonds and all of these different interactions determine the shape of the protein, which is why I said before that the sequence of amino acids was so important. And not just the sequence, but also what amino acids appear. So for example, if you had two cysteine amino acids in a protein, but then you took one of them away, and then you wouldn't get this interaction, and this bend, I suppose, over here might just end up not being a bend anymore and interacting with something over here, because it doesn't have that strong disulfide bond anymore. And changing the shape changes the function of that protein, which is why the primary structure is so important. Then we have the quaternary structure. The quaternary structure is where you have different peptide chains or prosthetic groups interacting together in conjunction, essentially. You also have different types of quaternary structures. You've got conjugated and non-conjugated. And essentially, non-conjugated, let's start with that because that's a little easier. is only made up of different peptide chains. So only peptide chains are present in a non-conjugated quaternary structure. But conjugated has prosthetic groups as well. Groups. And essentially what prosthetic groups are is essentially they're pretty much metal ions that are added onto the structure to help it perform the function. So for example, this is hemoglobin, and hemoglobin consists of 4 peptide chains, which are this blue and red, like helixes. And then these things over here are iron ions. And so those iron ions help the hemoglobin to perform its function. So that's the quaternary structure. Okay, so now we can move on to the different types of proteins, which are the fibrous and the globular proteins. And what we need to know about these is their function, or their general function, their structure, and different properties that they have. And it would also be good to be able to name some of them. So let's start with fibrous. So fibrous are usually structural proteins. They're very long and narrow. Because that helps them with the function of being a structure, essentially. But they are less soluble in water or polar substances, and they have a repetitive amino acid sequence. In comparison, globular proteins are usually catalysts. They are round and spherical. And the way I like to kind of remember which is which is because I don't know what it is, but the word glob just sounds like it would be round and spherical. So if you just remember, like a glob of ink, for example, would be like a general circular shape, general round shape. So that's how I think you can distinguish between them. They're generally more soluble in water, and they have irregular amino acid chains. So they're not repetitive in the ways that fibrous proteins are. So then types of fibrous proteins can include collagen, or keratin. I'll just label 2 for each. And then for globular, you have hemoglobin. I do not know how to spell that word. That's definitely wrong. Oh wait, was it right? Hemoglobin, and also insulin. So yeah, that's all that you need to know for higher level. It is quite a lot in my opinion, but once you get the hang of it, I think everything will turn out just fine. But yeah, that's all, and I will put all these notes on a whiteboard and then put it down below. Okay, thank you so much for watching.